Prophage and provirus are both terms used to describe viral DNA that is integrated into the host genome, but they refer to different types of viruses infecting different type of host. A prophage is the phage, bacterial phage genome that has integrated into the DNA of a bacterial host cell. This integration occurs during lysogenic cycle of the phage. Prophages remain dormant within the bacterial chromosome until they are triggered to enter the lytic cycle, where they replicate and produce proteophages, ultimately lysing the host cell. Provirus, in another hand, a provirus is a virus genome that has integrated into DNA of eukaryotic host cell, typically belonging to animal cells. Examples of such viruses include retroviruses like HIV. Similar to prophage, provirus can remain dormant within the host cell genome and undergo periodic activation to initiate viral replication, leading to the production of new viral particles. So the differences in latency of animal viruses and lysogeny of phages. So the latency of animal viruses refers to the ability of a certain viruses to establish a persistent infection within their host, often remaining dormant for extended periods before reactivation. This latency is typically characterized by the integration of viral DNA to the host genome and the ability to periodically reactive and produce viral progeny. Lysogeny of phages, on the other hand, refers specifically to the integration of phage DNA into the bacterial host genome of prophage and the maintenance of dormant state within the bacterial cell. This state can last for multiple bacterial generations before being triggered into the lytic cycle. The differences between lytic and lysogenic replication of bacterial phages. So the first one, lytic replication, the phage affects the bacterial cell, hijacks its cellular machinery, and immediately begins replicating its genome and producing viral components. The host cell is lysed, is broken open at the end of cycle, releasing numbers of progeny variants into the surrounding environment. Lysogenic replication, the phage integrates its DNA into the bacterial chromosome and become a prophage. The integrated prophage is replicated along with the bacterial DNA during self-division without causing immediately harm to the host. Two examples of where lysogenic conversion has contributed to pathogenicity: Vibrio cholerae. The bacterium Vibrio cholerae, which cause cholera, can become more virulent due to lysogenic conversion of the bacterial phage. This phage carries genes encoding cholera toxin, which is contribute significantly to the severity of the disease. Clostridium botulinum, the bacteria responsible for botulism, can become more pathogenic through lysogenic conversion by phages carrying genes encoding botulinum toxin.